Hello, everyone. Welcome to our panel discussion on Epigenomics Beyond DNA methylation. We're glad you're here. I'm Amy Lyndaker. I'm from the Epigenomics Core at Cornell University up in Ithaca, New York. Before we begin, we have a few housekeeping announcements. Please silence your phones. Photography at ABRF meeting sessions is permitted, unless the speakers request you not to. Recordings of the program sessions will be available on the meeting website following the annual meeting. I think that's about 2 months out. ABRF's social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post on social media. This is a discussion session. So once our sponsor talk has been completed, questions are welcome at any point during the presentation. Please do come up to the microphones to ask questions. We'd love to get a good discussion going and have your input. All attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. Also, hopefully you noticed that out in the lobby area, there is a board where you can suggest meeting topics and sessions for next year, and that's how this session came about. So last year, I came to the ABRF meeting in Minnesota. I noticed there really wasn't anything about protein DNA binding and histone modifications. All the epigenomics was kind of limited to DNA methylation, and so decided to start this session. I'm really glad that we're doing this today. So joining me for this session today, we have Alex Zevin from the Fred Hutch Cancer Center. We also have Abhinav Jain from MD Anderson Cancer Center. And we also have Aaron Wenger from PacBio, and PacBio is our sponsor for this session. So first we're going to have our 10-minute presentation from our sponsor, PacBio, and then we'll circle back and have an introduction to each of our core facilities before we move on to our discussion.
Great, thank you, Amy, and thanks everyone for joining the session today. I think it's a great topic. I'm excited to be part of it. So as Amy said, my name is Aaron Wenger. I'm a Senior Director of Product Management at PacBio. I've been at the company for about 8 years, and today I'm going to talk about how the PacBio long-read sequencing can be used for epigenomic analysis. So at its core, PacBio, as I'm sure many or all of you know, is a long-read sequencing technology that sequences individual molecules in individual wells on a sequencing chip. The key characteristics of the sequencing technology is that it produces long reads, all the way from a few hundred base pairs up through tens of kilobases. It's extremely accurate, producing reads that are free of systematic error, with most of the bases being Q30 or above. It provides complete coverage of the genome with no biases by GC content or things like that. And as I mentioned, it's a native molecule sequencing approach. So any epigenetic marks that are present in the DNA are also present in the library, present on the instrument, so you're able to detect them if there's a noticeable signal in the sequencing. So if you looking at many aspects of a genome with short read functional genomics, we typically would think of a variety of different assays, and I know we'll hear about some of those throughout this session. So you can have ChIP-seq or CUT&RUN to identify enhancers or promoters, DNase or ATAC-seq to look at open chromatin, RNA-seq for transcription, and then bisulfite sequencing or similar approaches to look at DNA methylation. With the standard hi-fi long-read sequencing approach, you get many of these omes in a single experiment without having to do additional preps. So the 5-methylcytosine, I know this session is focused on things beyond methylation, but just to start with what is standard, a standard hi-fi sequencing run on any of the PacBio systems will give you accurate reads with high base quality that can be phased into haplotypes, but you also get layered on that the 5-methylcytosine calls. That's just part of a standard run. And this is available on both of the PacBio systems that are available today, the Vega system, which is our benchtop system you can see at the booth, and then the Revio system, which is a system for doing hi-fi sequencing at larger scale. So the Vega system does 1 SMRT cell at a time, producing the 5MC and 6MA calls directly off instrument. Revio will do up to 4 SMRT cells at a time, producing the same data type. So just want to go through some of the ways that you can use hi-fi sequencing to go look at aspects of the epigenome beyond methylation. I'll start with the chromatin epigenome. And this is really reliant on, I'll actually feature a number of different approaches that were developed by PacBio customers. This one was developed at the Sturgacis lab. And Andrew Sturgacis is now a professor at the University of Washington. He developed this approach called FiberSeq. And the basic idea of this approach is to add a methylation mark to DNA that's not endogenously present in human genomes. So specifically, they use a 6MA methyltransferase, and you treat DNA when it's still in chromatin with this 6MA methyltransferase, and wherever that enzyme is able to access, it will add methylation marks to the DNA. Then you do a DNA extraction and a standard sequencing, and this methylation mark can be read off in PacBio sequencing at the same time as you're getting your base calls, the same time as you're getting your 5MC. And because it's not natively present in human DNA, you know that anywhere the 6MA was added, it was added through this experiment and not biologically. And this is showing on the right here how some of that data look. You can see reads aligned to a 7.5 kilobase region, and the green marks A's that are methylated. So you can see kind of a regular pattern of chromatin where you see every 150 bases or so, there's a run of A's and then a run of gray and then a run of methylation. But right at the promoter of the SRF gene, you can see a larger region of open chromatin. So that's kind of how this data looks. And again, this is layered on with the same 5MC and base calling that you would get in a standard run. And this particular methylation mark is called on instrument, so it's part of the standard output for both the Revio and the Vega system. There was a nice paper earlier this year that took advantage of this to produce what were called, in the paper, synchronized multiomes. The idea here was they took DNA from a variety of humans. They did both some genome in a bottle reference samples and then some patient samples. They did this FiberSeq treatment and also took RNA from the sample and did a long-read RNA prep. And then pooled all this together and sequenced it on a single SMRT cell in the Revio system. So you got out of the FiberSeq experiment, the genome, the CpG methylome, and the chromatin epigenome all on the same exact reads, and then you had the transcriptome from the same sample as well. And that produced data that looked like this. I mean, this is a nice locus to illustrate how this data can look. So here, the red and blue show the 2 haplotypes. So the reads are grouped by sequence. The transcripts are grouped by sequence. And you can see in the kind of brown colors that there's, that's the CpG methylation, and then the purple bar underneath that is the chromatin accessibility, and then the kind of blue bars show the RNA transcripts. So you can see in the first highlighted region that the top haplotype has low methylation, it has high chromatin accessibility, and that's at a bidirectional promoter, so you get transcripts that go in both directions. And in the bottom allele, that first region is methylated, it's silenced, it's not accessible, but the right promoter is accessible and you get that transcript there. So you can see this clear haplotype picture, everything layered on together, all from the single sequencing run. As a second aspect, I just want to talk about transcription factor occupancy, and this is another approach developed by Andrew, which is a similar technique to FiberSeq, and fundamentally it's taking advantage of that same idea where you're adding a mark and you have DNA that's protected by transcription factors or histones, but here it's an approach that lets you get much higher coverage by doing targeted sequencing of certain regions. So one shortcoming of the FiberSeq approach is it's based on methylation, so if you PCR it afterwards, the mark is erased. The idea of this deaminase-assisted FiberSeq is rather than using a methyltransferase, you use an enzyme that deaminates and turns C's into U's, and thus those base changes are perpetuated by PCR. So you can get much higher coverage views of, with the same fundamental data type as you get from fiber-seq, but just at much higher coverage. And this is showing really high coverage, thousands of reads of coverage of a promoter for this NAPA gene. And what you can see is the reads are kind of grouped by transcription factor accessibility. And what you can get in these reads that are 5, 7 kilobases long, you get which transcription factors are bound, the areas that are not read, and then which ones are not bound, which are shown in the areas that are read. So you can see things like co-occupancy and what percent of reads have one transcription factor bound and which ones don't. And that's kind of shown in the top there with those different motifs. I think that's a really interesting data type. Just as one last area, we also can use hi-fi sequencing to look at 3D chromatin structure. And the first example of this is actually, again, building on this fiber-seq idea with a method that was recently published out of the Altimos lab called FiberFold. And this takes in as input the fiber-seq data type, and then it uses a machine learning algorithm that's been trained on chromatin accessibility and high-C maps to infer the 3D structure from the local chromatin structure and phase and methylation. So the inputs to this are the chromatin structure and the methylation that comes off of those hi-fi reads, and the output is a contact map. Then you can also directly measure the chromatin structure with hi-fi sequencing using an approach called Sci-Fi, which is high-C with long-read sequencing. That's basically what the method is. You make longer fragments, you still ligate them together, and you sequence them on the long-read sequencer. And the biggest distinguishing factor here is that you're able to map into some regions that are not accessible with the short fragments that you get off of a standard high-C. So this is showing a contact map for long-read hi-fi sequencing or Sci-Fi on the top, and the same sample with standard short-read high-C on the bottom. So you see agreement, but then there's that region in the middle where there's no information from high-C, and you do get contact maps from Sci-Fi. Yeah, so I think in conclusion, the hi-fi sequencing that you get off the Vega and Revio system is able to provide really rich epigenomic information all in a single assay. This includes in any sequencing run all this genomic information plus 5-MC methylation. And then with these the additional assays, you can get the chromatin epigenome, you can get transcription factor occupancy, and 3D chromatin structure. So with that, yeah, thank you again for the time, and yeah, really appreciate the opportunity.
Thanks so much, Aaron. So we're excited to be here to share our knowledge of core operations that are specific to epigenomics assays. The assays and services we're going to focus on today look primarily at protein DNA binding genome-wide. And so this includes things like ChIP-seq, ChIP-exo, CUT&RUN, and related assays. I thought since this is a discussion session, just start out, see where everybody's at in the audience. So how many of you have at least heard of these assays? Okay, good start. All right, how many of you work in core facilities that offer one of these assays? Ooh, a few. Great. How many of you get customer questions, like do you offer these services or requests to offer them? Great. It seems like there's a need there. How many of you then would like to offer these services but maybe need resources or knowledge or, yeah, great. Maybe we can all help each other get there. So now we're going to each spend just a few minutes introducing our core facilities and what we do. And then we'll open it up to questions from the audience and amongst the panel members. So I'll get us started. Again, I'm Amy Lyndaker. I manage the Epigenomics core at Cornell University. And I'll just mention we are separate from the medical school that's in New York City. We are up in Ithaca. We're part of the Biotechnology Resource Center, which is within Research and Innovation section. And I should also acknowledge my funding. I had a travel award from NeuralSkid, the regional chapter. Much appreciation for that, helping me to get here. So this is our staff in the Epigenomics core, what we call EGC. Our director is Will Lai, and he also does all of our bioinformatics. I manage the wet lab biochemistry side of the lab, and then we have 2 technicians who do most of the bench work. If you want to contact us, I left our information up there. Since we're part of the BRC, it's brc_epigenomics@cornell.edu, or if you can just email me directly or catch me at the meeting, I'm happy to chat further. We primarily offer an ultra-high resolution version of ChIP-seq that's called ChIP-exo. And I'm going to circle back to this in more detail later, but I'm just throwing an example up here for now. You can see this is a plot of the reads from a ChIP-exo experiment, and it is centered on the motif where this protein is known to bind. And you can see these sharp peaks on either side. It's basically showing you the footprint of that protein on the DNA at very high resolution. I'll show this again later, but there's a -10 and +11 base pairs on either side of that motif. It's very precise, and it's a stranded assay. It's a really nice way to look at where proteins are on the DNA. We do also offer ATAC-seq, but I'm not going to talk about that today. So we do offer end-to-end services. So clients give us cell pellets or frozen tissue, and we give them back their sequencing data. We do all the bioinformatics in-house and all the processing. We primarily take in mammalian cell line samples, but we do take in tumor biopsies, mammalian tissues, sorted cell populations. We do yeast experiments, take in bacterial pellets. If you have the yeast tap tag collection or something like that, we're happy to use those kinds of samples. If you have something else, we do take in, we've done fish brains, butterfly wings, whatever you have, we're willing to work with you to get those samples done, and that keeps it fun. You know, we primarily offer this one assay, but it's always different. We process about 4,000 samples a year. We do take in external clients and internal to Cornell, so people send us samples from all over the world. We do all of our sequencing in-house, so we have an Element Biosciences Aviti, so as soon as the technicians are done with their library prep, I can throw it on the sequencer, and then we start analyzing the data right away. So it's really nice end-to-end. We have control over all the parts of the process, and some of that is relatively new. When I started, we didn't do our own sequencing, and it's been really nice to have our hands on it the whole time. So we have a custom LIMS system that keeps us all together. It's called PEGR, Platform for Epigenomic Research, and it's shown here. This is where we integrate all of our metadata, and I love PEGR. It's the best thing ever. This is open source. I can give you the link to our GitHub if you'd like to use it. Highly recommend if you don't have a system in place like this to do it. So this is where we keep information about our protocols, our sample metadata, so when did this sample arrive? How many cells are in this sample? What kind of cells are they? Which antibody did you use? What's the lot number on that? Everything is integrated in here. Which sonicator did you use? And then this is all linked to, we do our sequencing. Our sequencing information gets incorporated into PEGR, so it's integrated with the Galaxy, and then we can use this to deliver our data. So we do peak calling, we call motifs, and then we can output data that we can show to clients to generate the report. So we have, just not tall enough to see the screen here. Our technicians produce a QC report once the experiment is finalized, and so they can include tag pileups, some of the sequence stats, write output directly from PEGR, and then we can also pull FASTQ files, BAM files, stranded bigwig files right from PEGR to do our data return to our clients. So it's really nice to have this all integrated in one system. And I'll say, if people ask, hopefully we will talk about antibodies later. If a customer comes to us and says, hey, have you ever chipped, protein X? We can look in PEGR and say, okay, when have we chipped that target? Which antibodies have we used? And the data is all right there. So I can say, yes, we pulled out the motif with this antibody, but not with this one. Or this lot number didn't work, but this one did. We used this many micrograms of that antibody. So it's really informative for us when we're helping our clients. I guess I'm going to have to go quickly here. So just a little bit more information about the assay before we move into the rest of the session. So ChIP-exo is a very high-resolution version of ChIP-seq, and some people are familiar with ChIP-seq. Basically, you have a protein that you're interested in, and it binds to DNA. You can use an antibody to recognize that protein, And then you capture that DNA and make it a library and sequence it, and you can find out information about what motif that protein is binding to. But in general, ChIP-seq is kind of not very high resolution. And so what Frank Pugh's lab did when he was at Penn State, he's now at Cornell, is to add an exonuclease step in that process. So the exonuclease chews the DNA such that the 5' end of the read is now at the boundary exactly where that protein is cross-linked to the DNA. So you can get very precise footprint of where the protein is located. So you can see these really sharp cartoon peaks here. And then I'll show that example that I showed before of the USF1 transcription factor binding to DNA. It's known where it binds. It has this motif, CAC-GTG. And so if we plot the reads from a ChIP-exo experiment of USF1, the top strand on the top, the bottom strand on the bottom, you can see these really nice sharp peaks at 10 bases before and 11 base pairs after, delineating that really nice footprint on the DNA. Let's say we have automated the ChIP-exo assay for the Epimotion 5075T, so we can do high throughput as needed. We do 96 ChIP-exo samples at a time on the system. So if a customer wants to do chip validation of antibodies, just completed 2 projects of 600 antibodies, we can do that. You know, it's 7 runs on the Epimotion. So we can do projects as small as 2 samples up to hundreds. And so that's how we operate our facility. Now I'm going to go on to the next speakers to give how their labs work, and then we can have some discussion. Next.
Hi, everyone. Thanks for coming out. My name is Alex Zevin. I'm the director of the Genomics Shared Resource at Fred Hutch Cancer Center, and thanks to Amy for inviting me to present today. I'm going to talk a little bit about the cut-and-run and cut-and-tag services that we offer. But brief overview of the Genomics and Bioinformatics Shared Resource. We support the full life cycle of genomics projects, starting from consultation and experimental design, technology selection, and all the way through library preparation and sequencing and bioinformatics analysis. It's all integrated together to provide the best outcomes for our users. And that QR code will take you to our website on the Fred Hutch site. I don't know if I put our contact information in here anywhere, but we're genomics@fredhutch.org if you ever want to get in touch with us. We have 8 people on our lab team and 5 now on our bioinformatics side. And we support a full suite of library prep and sequencing technologies. I'm not going to dwell on any of these in particular, but we do have a pretty robust fleet of technologies and services. And the ones that I'm, of course, going to draw everyone's attention to today are our auto cut and run and auto cut and tag services that we developed in collaboration with the Hennekoff lab, who was the inventor of these technologies. These are both fully automated chromatin profiling methods to look at occupancy of whatever transcription factor, histone mark, genome-wide that you may be interested in. The way that we provide these services is that users will initially contact us for a consultation to discuss the scope and aims, and that's when we'll help guide them towards one method or the other. For those of you that aren't necessarily fully familiar with these, cut and run is cleavage under targets and restriction using a nuclease. This is a protein A conjugated eminase, and cut and tag is cleavage under targets and tagmentation, and that's a protein A or protein AG conjugated transposase. Cut and run tends to be a little more robust for like low abundance marks, for example, transcription factors, whereas cut and tag is much more sensitive. You can get away with many fewer cells as input, but it often works better for histone marks or transcription factors that are binding the DNA directly rather than transcription factors that might be part of like a complex. The way that we offer the service is we accept samples that they're bound to the antibody, and they are bound to the magnetic CON-A beads. This is because we do a lot of volume, and we can't really afford the time to take every project and optimize sample prep for every individual use case. People are bringing all kinds of cells, all kinds of targets from all over the country. And so, we'll provide significant support in optimizing that sample prep, but we don't directly do it for anyone. Users need to schedule their runs in advance. This is because of the high volume that we do. It also gives them a chance to work out sample prep up front of their runs. If they need to push things back, no problem. We're always happy to provide that, or to accommodate their schedule changes. So once someone hands us a sample that is bound to antibody, bound to CON-A beads, we take over from there. We'll perform the library preparation starting from whatever the digestion method is, if it's the tagmentation or the MNase digestion. We'll take that through the remainder of the library preparation, sequencing, typically now on our NovaSeq X Plus, and bioinformatics analysis, which always includes basic data QC, alignment to whatever reference genome, and then peak calling using a couple of different peak callers, and we'll just provide those data in bigwig format. We offer 24, 48, and 96 sample tiers. The 24 sample tier is specifically for folks that are trying to work out sample prep optimization and want to maybe run like a panel of different conditions and not have to pay for a whole plate, basically. So we did that, we introduced that tier intentionally to try to help facilitate folks that might want to validate using the library prep method that they would ultimately run the full service on. So some of the things that we do to ensure the quality and consistency of every run, because we're not doing the sample prep, so we kind of need a little bit of insurance for ourselves, is we always put process controls on every single run. So these are K562 cells that we've done the sample prep on, and we've optimized that, and we have a panel of targets that I've listed there that we run on every single run. So if you ask for 96 samples, you're actually getting 92, because we need those 4 wells. Sometimes we'll do an IgG control, but it's always blank, so we don't want to waste the time every day. We also do routine testing of new enzymes and reagents from alternative suppliers to try to manage costs and improve data quality. So, some quick examples are we updated from single indexed adapters to unique dual index UMI adapters. That's really good for some of these transcription factors where you wind up with super high duplication rates. Helps you get rid of some of those PCR artifacts. And we've also sourced lower cost T4 ligase and PCR master mixes to, those are kind of some of the expensive parts. We're lucky, we touch the Hennekoff lab directly. They give us the protein A conjugated MNase and protein A conjugated transposases. So that really helps to reduce costs. So I hope that Steve sticks around for a long time. And we're always refining our methodology to improve yield, especially from low abundance targets. So for a while, or as the service started, we were using digitonin to permeabilize cells. We were getting feedback that users were not getting very good signal, especially from MYC antibody. And so we did a bunch of rework on the assay and development work to try to improve that signal. And what we eventually wound up doing was switching from digitonin to Triton X100 to permeabilize the cells. And we also increased that MNase digestion time and temperature, and that helped us recover signal for those low abundance marks, as well as really blow out the signal for histone marks and things that are high abundance. And the plots there are showing the performance of our process controls over time, right? So we've done a bunch of these runs, and so we can look at things like duplication rates, number of peaks called, and consensus peak recall rates for every single mark across time, and that helps us ensure that we're getting consistent performance. And it also helps us when users come to us and say, hey, my MYC antibody isn't working anymore, we can say, well, ours is, so maybe you need to go revisit your sample prep, or we can look at our data and say, oh, well, yes, something is falling apart here. So this is good insurance for us and good monitoring. We also do active method development. So Steve's lab recently published on the cleavage under targeted accessible chromatin method for FFPE samples. And so we're actively working to develop this out into an automated service. This is a really robust method for profiling the loading of a paused RNA polymerase II on chromatin. It gives you data type that is very similar to ATAC-seq. But this is compatible with FFPE tissues. We're showing that we're able to recover signal from fixed cells as well as fixed FFPE tissues from mice. And we're working on some data analysis, and we're also showing that we can do this in frozen cells as well. So building out new services to help provide these high throughput methods to the community. And that's all I have.
Good morning, all. First of all, thank you, Amy, for organizing such an important session. I'm new to ABRF. This is my first time. And thank you all for coming. I have an active lab at MD Anderson. My research is on long non-coding RNAs, but my other hat is to co-direct the epigenomics core, and that is what I'll talk about. So those are my credentials down there on the slide. So I think before I tell you a little bit about my core, I have a quick recap about how the epigenetic landscape control all the information that we are after, the gene expression in different states. So these are major highlights, how the DNA loops and how the chromatin marks, like histone modification as a LX, and how different proteins, as Amy has referred to, are interacting with the chromatin, not DNA alone itself, and regulate the gene regulation program. So you can see in those boxes, we have categorized into 4 different portions of the mechanism that regulate epigenetic landscape: the protein-DNA interactions, the DNA modification, the RNA and protein interactions, such as long non-coding RNAs, which is focus of my lab, and then higher order chromatin organization. So I think the focus of this session is the protein-DNA interaction. So I'll show you the core, actually, the epigenomics core at MD Anderson is run by me and my colleague, Dr. Marcos Estacio, whose leadership is on DNA methylation, and rest of all the stuff is under my purview. So our mission, when we started this core a few years ago, was to facilitate basic and translational epigenomic research by providing expertise and access to cost-effective advanced assays to investigate DNA-protein interactions, chromatin organization, and DNA methylation. And to be honest, when I started it, when I was involved in it, our goal was only to do ChIP-seq for people, but then over the years, we cannot just sustain by ChIP-seq. So there are a lot more components of epigenomics that we actually, as you will hear. So that was a short team. So this is just a project workflow, how we have set up. So there is initial consultation with the users. And that consultation is very scientific. That's where we actually design the experiments for them, because MD Anderson is a huge community of both basic and clinical scientists. And we don't just serve MD Anderson. We serve the whole Texas Medical Center. We have a lot of clients across USA and even in UK. So once that initial consultation, we design what kind of assays to be done. We give a very strict recommendation of what kind of sample preparation the user has to do, go back in their lab. Then the samples are received in our lab, and those assay-specific steps are done. We prepare the library. We have very stringent QC, as you would see in my subsequent slide, at every step, to ensure that we don't waste time on sequencing and post-sequencing computational analysis. We are not an NGS core. MD Anderson does not allow to have sequencers outside the NGS core, so those samples are gone there. Then there is the FASTQ files come back either to us or to the lab itself, where they have computation analysis capability. And I'm a very hands-on person. My team is very hands-on, so we are involved in various aspects of the project, not just processing the sample. So there are a lot of labs where I sit on their lab meetings to discuss the data, and even then you get that kind of effort in their grants and manuscripts. Okay, so these are some of the services which actually over the years have been expanded under my purview. So the idea started with just doing ChIP-seq, and we do different versions of ChIP-seq. We do in the high throughput format in the 96-well plate versus a more traditional way in the tubes as well. And the ChIP experiment is not just the sequencing, but there are a lot of labs who just want to know, hey, this protein A, protein B, does it bind to my gene of interest? Because this is what I'm noticing with my other assays, so we'll help those. I'll show you a couple of examples. We also started recently doing the higher chromatin organization, like high C and high ChIP kind of assays. We also just actually optimized our assay to do actually ChIPs from liquid biopsies. We have validated for several histone marks. And as Amy also mentioned, we have assays for ATAC-seq. And we have actually optimized the bulk ATAC-seq assays to go to very least number of cells. And in development, we are actually now developing spatial ATAC-seq and spatial cut-and-run. So this is the chromatin side portfolio of our core. And we use this actually as a scientist because epigenetic code was the word which Dr. David Ellis actually started. So we thought that this would perfectly fit the phrase that use epicode to delineate the epicode. So this is just the workflow and the sample requirement. So you can see there are 3 assays pertaining to today's session. So for ChIP, we accept all kinds of samples, frozen, cross-linked cell press pellets, flash frozen tissues. And MD Anderson has plethora of tissue sections on slides. So we have even validated our protocol to actually have those people go back and scrape the tissue from their slides and come back to us. And then we did ChIP from that. And as it says, plasma from liquid biopsies. We also do a lot of cut and run and accept fresh or live cells because, as you may have heard, and Alex can tell you more about that, the viability of the sample is the most critical part for a cut and run success. We accept cryopreserved or cross-linked. Again, depending on the antibody, it's a very customized assay. One shoe does not fit all, and even on flash frozen tissues. And we do all these steps in my lab. And if you see, I have highlighted QC, so which is the most critical part. We spend a lot of years actually coming up with those QC statistics so that we don't have those kind of feedback from the users that, hey, it didn't work. So we want to make sure that we have as stringent QC steps as possible. And those samples then go to the NGS core, and then I'm involved in a lot of post-processing consultations. I'm on a lot of manuscripts which come out from these labs, and I help them write their grants and even the data interpretation. Just a couple of quick examples. So this is an example of a paper. So these, Dr. Raghu Kaluri's lab, they had developed a KRAS induction model. And all they were wanting to know is that whether this gene of interest, the chromatin state of this gene of interest was changing. They didn't need to do ChIP-seq and all that stuff, so they came to me, and we designed a ChIP qPCR experiment, did a bunch of histone and non-histone modification marks just at few locus, and that paper was published. So we just saved them a bunch of effort by just doing ChIP qPCR, because I remember the initial question they asked me, can we do ChIP-seq to determine what happens to fast genes? So that is not the answer. This is another paper which actually came out a couple of years ago. Is this lab actually has developed a very unique mouse ES differentiation model, and we're looking for a bunch of histone modification and ATAC-seq. So since my lab portion can do all of them, so they didn't have to prepare different samples for different assays. So we coordinated with them so that in one go we could do everything for this lab. This is cut and run. Alex mentioned some benefits. So cut and run is a completely different technology which was developed by Dr. Hanikoff. And these are a couple of advantages I have listed here. You use less material, starting material. You can get more much better signal to noise ratio. So we actually, based on the initial communication with the users, we decide which assay is best for them. There are a couple of examples I'm stating. This is a friend of mine. He works in Texas Heart Institute. they developed this system of isolating cardiomyocytes from mouse, but they could never do the chip because they didn't have enough material. So that is one place we actually optimized CUT&RUN assay to fit their cell line, and the results were already published. This is another report from Dr. Sharon Dent, who's one of the very famous epigenomic person. So their lab works on these big chromatin remodeling complexes. So although you would think that those remodeling complexes, it would be easy to actually identify their genomic landscape, but it's not. Some of these are the chip cannot reach the antibody to actually isolate. So they struggled a lot. We actually modified our CUT&RUN assay to be able to pull down those complexes, and that paper was published late last year. So that is that. I'm happy to have discussion. This is our group. On one side is the chromatin group, on the other side is the DNA methylation group. We enjoy the institutional support from our institute, and also we have a very active advisory committee, which includes a lot of scientists and admin support. So just to make what we are doing is worthy for everyone's interest. So happy to join the discussion, and thank you again. So.
Thanks. So now we're going to open it up for discussion questions. So if you have questions, please do come up to the microphone. I'll get things started. I have a lot of ideas, but you guys can jump in. To start, do you have certain antibodies that you recommend? Do you have a list of approved antibodies, or can clients bring anything?
Yeah, is this on? Yes, thank you. Yeah, we do keep a list of antibodies, like you had mentioned, that we've tested before and have had success with. We keep in that list, what dilution factor they were used in, if there was a secondary antibody applied. And then we also tend to point people to, like Epicipher has lists of validated antibodies for CUT&RUN as well, so we'll point folks to that. If it's a novel target, then we defer to the optimize your sample prep pathway.
In my case, I'm a scientist myself. I don't want to lose any money. So anytime if someone comes to me, I have one of my technicians, his first job is to look at PubMed of published papers on that particular transcription factor and figure out what antibodies were used if this is new to us. And if it is, we have already succeeded with something, we keep an inventory of those antibodies, and then we refer our customers to those. And we, as you said, you have a good LIMS system. We also have a different kind of our homegrown system, so we keep a very strict inventory of conditions of antibody used. So, yeah.
Go ahead, Stuart.
Hello, Stuart Levine, MIT. With these end-to-end services, one of the challenges I'm curious your thoughts about is as a bioinformatics core, one of the things we're always thinking about is that if we do analysis for somebody, they are less hireable as a faculty member because they can't answer their own questions about their own data. As an educational facility, in addition to a shared resource, how do you square that circle?
I'll say, for us, we do expect the clients to analyze their data themselves and produce the figures themselves, but we do some minor analysis just to say, this is a good experiment. So we show them our controls. If we know that their target has a specific motif, we'll do a tag pileup and say, look, it worked. But we're not expecting this. We don't want them to publish our analyses. We want them to analyze it. So I guess one of the questions I want to come back to later, we can do it now. Clients always come up and say, so now what do I do? Here's the data. So now what? But we're not going to do we don't know their scientific question the way they do. So I have zero expectation that we are going to analyze it in the way that they want. We are providing their data, but we do some analysis. I don't know what other labs, yeah.
Yeah, we get a lot of people that want to do cut-and-run, cut-and-tag who aren't necessarily bioinformaticians. So we don't necessarily expect them to be able to do the alignments and read pileups all by themselves. So that's where our bioinformatics group comes in. It is a highly interactive service. So, you know, this is not done in a silo, right? Like our bioinformatics team is working with the investigator to, you know, understand the experimental design and make sure that the data we're providing back is helping to support whatever hypothesis that, or, you know, reject whatever hypothesis that they may have been working on. Ultimately, the biological interpretation of those results is up to the investigator. We don't really do much of that.
I can answer in a different way. So we usually, in our initial consultations, our goals are set in the initial consultation. If the bioinformatician is doing certain things, this is the expectations. And there are certain labs who don't know what to expect. And that's when I get on those meetings and I draw some lines. You know, this is, because bioinformatic analysis, for all who do this, you can imagine this is an open-ended process. It would never end. Now I need my graph this way, now I might need my graph this way. So you have to, yeah, I totally get the pain. You have to draw lines somewhere. So that is actually, at least I do, yeah.
Oh, you're going to answer my
I was
question.
going to add, yeah, we also provide resources for.
I understand the resources,
Yeah.
but again,
Yeah.
my concern is that you're weakening the postdoc.
Yeah.
You're weakening their ability to read a manuscript as a reviewer and critically evaluate that manuscript. You're weakening their ability to write a grant and know that those algorithms are going to work.
Yeah, they need
And
to
so
understand
it's,
it.
it's, and again, I run a bioinformatics course.
Yeah.
This is one of those major paradoxes. And
Yeah.
it's one thing for clinicians, like their job is to do surgery. They're not going to be evaluated as a bioinformatician. But I'm going to let other people. But I, as an informaticist, that's something that's always in the back of my head.
Good question, actually.
Hi, Claudia Lalancette, University of Michigan Epigenomics Core.
Cool.
Nice to meet you all. So I'm going to start by answering what I think Stuart was looking for, especially on the perspective of the students and postdocs.
Yeah.
I think it's more of a hands-on training understanding of the technology and how the samples are processed so they can have a better assessment of critically understanding how the sample was processed and like the nature. So at the core, I am starting on the CUT&RUN. I've been on the back burner. I've been doing ChIP beginning till end, pretty much like you. After 5 years of doing consultations, of explaining to students and customer, researchers where their samples were shite. And there was no point in doing libraries for it. So I preferred putting the investment and the time into taking their samples in. I do have, I kind of educate them on it too, and just do the optimization. I think it's a better time, better usage of my staff and my time to do it from beginning till end. I think you probably figured that one out too, right? I've had a lot of demand of students and postdocs to come and be trained, and I'm actually starting a service. I'm implementing that. I will give them training on how to do it. In some cases, they'll figure out that it's way too complicated for them, but at least they will have the knowledge. And I do a lot of consultations back and forth, and they ask me, and I actually send them to read papers because they are students or postdocs and they need to learn. So I don't know if that answered your question. Hopefully we can discuss it. My question is more for the CUT&RUN, and you're taking the cells are attached to the beads because of the optimization, right? Mm-hmm. So there's a simpler, it's just you do a nuclei isolation, because my understanding is usually the optimization is mostly on the digitonin that you need to permeabilize the cells. You totally bypass that if you do a nuclei isolation and you start with that for your CUT&RUN. And I think that's what you have to start with de facto with cut and tag anyway. So you would probably save lots of time and hurdles going that route.
That's good feedback. We haven't really encountered any problems starting from cells, both for cut-and-run and cut-and-tag.
I think you're lucky because you have the expert next door.
Yeah, and well, like I said, we're maybe a little spoiled with it with the Hennekoff lab around, but we have had folks from all over the country send samples, cells that are bound to antibody and in the permeabilization buffer and bound to the Con A beads from Michigan, right?
Cool.
So, the system works.
And I'll talk to you after.
Yeah.
Thank you.
I can actually just point in that question. So we actually, in my lab, we keep inventory of every system, every cell line we get, and what kind of buffer was used. So just in case if someone sends a cell line or a tissue like that, so we can just quickly refer back to our notes. Yeah.
Under which circumstances would ChIP-seq still be advantageous compared to CUT&RUN?
There are a lot of actually, we encountered that a lot. So a lot of antibodies, even though they are listed, they would work for CUT&RUN. Actually, in our hands, we have not had that kind of reproducibility as people would say that, this antibody is CUT&RUN valid. So blanketly, it would not work. Like this example, one of the example I showed, I did not show that example. I showed in an inverse way, but we have had, like for histone marks and all that stuff, CUT&RUN would work. But for a lot of chromatin remodeler antibodies, you may still want to cross-link a little bit and have ChIP-seq. And also, it also depends on tissue type, tissue or sample. Alex can talk more about it, because we get a lot of these from repositories. The samples have been stuck in freezer for years, and nobody knows how they were stored. So the quality of that sample actually may not be appropriate to do CUT&RUN, because the viability of the cell and the intact nuclei you might not get out of it. So that is one way I would definitely do ChIP-seq on those samples. Yeah.
I'll add also, it's not specific ChIP-seq, but for ChIP-exo, we can detect secondary interactions. So if a protein binds to the DNA, but you want to chip the protein that binds that protein, we can detect that, and we can look at the structure of that binding complex, where I'm not sure that would work if you didn't have cross-linked cells to start with. So.
Yeah, that's right. I mean, CUT&RUN does have those kinds of limitations to it, and we don't offer a ChIP-seq service in our lab. So, we'll always ask people to do small-scale experiments up front before moving into the larger scale automated service to make sure that they are like getting some signal out. You can just measure it by tape station, right? Are you getting library? And if they can get themselves to that point, we can feel relatively confident that we'll be able to carry out on the larger scale.
So Suzanne Sandmeyer, UC Irvine Genomics Core. So to refer back to one of the early questions, our bioinformatics team gives regular workshops because we've taken surveys and we know about a third of the people want to do it themselves, a third of the people want the core to do it, and a third of the people want everything. And so it's an important aspect of training and our service that we provide postdocs in the labs with some of the basic starter tools, and a lot of it really amounts to self-learning, so getting started is the key step. So, but my real question is, you guys have all done amazing amounts of technical development, and we find ourselves stretched very thin because of our recharge model for affording the time to do some of that critical development work. So how do you guys think about balancing recharge development activity in your budget?
I would say we have benefited greatly from working closely with academic labs who are doing a lot of assay development. I'm not the only one.
Yeah, same thing. We work really closely with Steve's lab still on, the CUT&RUN method is a great example of that. We're working actively with them. And we just carve out time where we can to get some production runs or test runs of the method in our lab going to the extent we can. So it's slow progress.
We do budget in some time for assay development, but as you know, it's hard to fit it in, especially if you get a lot of samples in. But yeah.
It's actually a very good question. So in MD Anderson, the central committee, so we just became institutional core a couple of years ago, but before that, the committee used to have these pilot grants where a core facility can just write a proposal, get like few $1,000s just for those assay development. So we actually kind of benefited from that for one of our assay development. But recently that thing has not been going on so well. So I did talk to our authorities because for all these new assays that we are developing, we are just actually just pushing money from here to here just to adjusting our budget, but it's not fair actually for the core and the core leaders to actually come up with their own bucket for money to do this. So yeah, I hope we have a solution, especially at MD Anderson, but we used to have a very good system back then.
Thank you, super helpful.
Yeah.
Other questions? How about sample QC? So there are a lot of stuff. I really appreciate that you underlined QC everywhere in your presentation. So maybe starting with samples that come in, how do you decide that they're good enough to proceed? And then I know, like later on your slide, you had a go, no go decision point. So are there, how do you define those?
I mean, like I said, we don't really work with the samples themselves, but insofar as library QC, we do tape station on everything, and if it produces a peak, we'll normalize everything to equal molar and pool that together. If there is no peak, often what we'll do is reach out to the investigator and we'll say, hey, this library didn't yield anything. What do you want to do? You can proceed, you can sequence it, maybe you'll get some reads, but it's not going to be much, or you can omit it from the pool. Oftentimes, they're already invested, so they say, hey, put it in and we'll add as much of the library as we have, and in it goes, and they get the data out that they get out with, the known caveat of,
Might not work.
be skeptical of your data.
What about at the beginning? So when they say, they're giving a certain number of cells, do you count cells?
No, we do not count cells. We give recommendations. We ask for 100,000 cells for cut and run and 30,000 for cut and tag.
And how often do they actually give you that number?
As far as I know, not all the time. But usually they can hit it, especially for folks that are working with cell lines. Clinical samples, people will put in what they can afford to give.
I think this is where we suffer a lot. This is the first pain point for any core facility to actually do the sample. So in the audience and whoever has done chromatin IP assay, you could actually vouch that the cross-linking is the most critical step. So we have a very specific guidelines how the samples need to be prepped, and we even have aliquots of formaldehyde and glycine to give away for just to make sure that there is a for labs in MD Anderson, hey, please don't worry about any catalog number of formaldehyde, come pick up from us, just to have that kind of linearity in sample prep. Now asking about, do they provide enough material? 80% of cases, no, because they are giving their best guesstimate on those numbers. And we have a method actually whereby the amount of DNA, you can actually calculate how many number of cells they gave us in the beginning, but we try our best. That's why the initial consultation is very important, is actually to tell the postdoc, graduate student, or even the technician from that lab that this is the very critical step. So please ensure to deliver the samples as required, but doesn't happen in 80% of cases, yeah, so.
And it's not really a formal in-process QC point, but we do quite a bit of visual inspection of samples as they're going through the processing.
Yeah, that can be huge. Does this pellet look right?
Yeah, exactly. Are we seeing an accumulation of beads in the waste or other debris in the waste vessel? Are you, is the sample like flocculent or anything like that? So just some general visual checks to check sample quality.
I'll add one more thing. So the one thing which we actually do in our core is, maybe at least I do, is there are 3 points in the process where a report is developed, and that report is shared by the PI. Those are the go, no-go steps. And they would have our interpretation that if we proceed with this sample, you are risking it or you are not risking it. So one of the, especially for the CHIP project, once we make the chromatin, that's the first QC step for us. We would know by the amount of chromatin that it's not enough to pull down this particular target protein. So I would go back and discuss that with users. Hey, we don't think, but if you really, this is the only sample you have got, we can give it a shot, but don't count too much on it. So those are very critical. I think those QC steps, and that also validates whether the sample quality or the amount was enough, actually.
Yeah, we ask for 10% more than we want, but we also request that they submit everything in triplicate if they can. So for cell lines, they often can. So we'll, take in a tube of 30 million instead of 10 million cells. But sometimes it's actually not even close to that cell number. And with tissue, it's limiting, so we'll just move ahead with what we have and explain the risk. Before we go. Go ahead.
Hi, thank you. I'm Shannon Hosu. I'm the director of the Bioinformatics Core at Harvard School of Public Health. I had a question about the spike-in controls when you do these studies, and what your experience is with them. Do you always do that normalization? Often we've decided not to include it, and I wondered just what your experience is and what your recommendations are.
We don't do spike-in controls. Everything gets normalized to the IgG. But that's a specific case, so.
We offer spike-in controls of yeast DNA to anybody that wants it. Sometimes they do, sometimes they don't. We often, if people haven't done a cut and run or cut and tag experiment before, we will say, hey, you need to run an IgG control to normalize for background. The other thing that we have, which is maybe not really an official control, is the MNase and transposase purifications that we get from the Hennekoff lab are not totally pure. So there's some background E. coli DNA as well that we can use, at least within any batch of transposase or MNase. We can use that as a pseudo spike-in.
Same thing for us. So for cut and run, we kind of use E. coli spike-in based on, but it also depends on my communication with investigators that whether you need it or not. But IgG is mostly used as control, so we always use IgG. And for CHIPs, no, there is, although there are spike-ins out there for the histone marks, you can have a recombinant nucleosomes, you throw it in there. But our bioinformaticians has given us feedback based on those spike-ins that it's more confusing than, rather using input or IgG as a control. So yeah, there is a lot of gray area there. So good question.
Yeah, that's been my experience as well, that sometimes the spike in normalization obliterates the signal.
Yeah, it just throws.
In trouble.
Yeah.
I wanted to just say also that in terms of education for postdocs and students, we do have open source materials that are quite comprehensive on chromatin biology. So I'd be happy to share that and for people to use that. There are flipped classrooms where there is a lot of self-learning to that as well, and so there's material out there as well if you want to use it.
Great, and we'd be happy to share. We have tutorials that walk through different processing steps that we could share with people if they're interested also.
Thanks.
Other questions?
Hello, my name is Charles Brandt from the Indiana University School of Medicine, and this is somewhat tangent to what we've been talking about, but do any of you have any difficulties or strategies that you use for data management and delivery? I'm guessing, or any pain points with that? Wondering if the Pegger system that you mentioned, Amy, helps with that, or any comments on that?
I would say, unfortunately, Pegger doesn't help us with that, and with our increased output recently, the storage has become a problem, and I don't have a great solution for that.
Yeah, same problem. I mean, the way that we do it is we'll dump FASTQs into the user's local storage on our HPC cluster, and then it's kind of theirs, and we'll push the analyses there as well. As they iterate through analyses with our bioinformatics group, that all gets updated, and you know that is essentially our data management strategy. Insofar as capturing some of the metadata, we really rely on the investigators to do that. You know, we can take in bits and pieces of it, like the antibody and then maybe the cell lines that were used and any specific conditions of the cell treatment, but you know, the minutia are kind of their responsibility.
Makes sense.
How long do you store the data?
We store our data, like we will hold data for 30 days. That's our policy. We wind up holding it for a lot longer most of the time.
Same.
And we give them, you know, we give users the warning, like it will be irretrievably deleted eventually, right? And so, you know, but once it's delivered to them, it kind of becomes their responsibility, and we'll delete it when we get around to it.
We don't generate big NGS files because we are not NGS core. We do have our MySeq and nanopore instruments just to play around, but yeah, not our problem. Our problem is the freezers, where we store those libraries and all that stuff. So we do have a timeline, but nobody wants them once they are sequenced and papers is published. So, but we still keep, I like to keep them for at least 6 years, you know, so yeah. And also most of our stuff is high throughput, so those are stored in plates rather than tubes, so it's easy, you know, more.
Yeah, that's a good point. The physical material storage, I mean, we really try to foist that off on the investigators as well. You know, we'll hold it until the sequencing results are complete and the data are all QC'd and look good, and then we will retain for 6 months. We'll tell them, hey, you know, if you want to retain this material, you need to come get it. Otherwise, if 6 months has elapsed and they haven't come to get it, that gives us kind of carte blanche to throw it out, you know, again, whenever we get around to it.
Thank you.
Thanks.
Angie Matheson from the Medical College of Wisconsin. Back to the IgG question. To me, it seems like you're trying, do you take them all the way to sequencing, or is it an internal QC step? Because it seems to me like you're amplifying something that's not supposed to be there, and a lot of times we have to go to a huge amount of amplification to get it on a sequencer. Are there, in the original ChIP-seq protocols, there was an input that just kind of told you about the whole ability to sequence the genome. Are any of those instituted by your institutions?
I can answer that question, Start. I'm sorry. So for, this is a very important point you brought. So we actually, as an insurance, for every assay we do and the integrity of the sample, we have a positive and negative control antibody for every ChIP assay that we do from a sample, every sample. So those IgGs, not all of them get sequenced. So for ChIP, we don't use IgG as a control. As you said, input is the control. For CUT&RUN, we use IgG as the control.
But that still seems biased on CUT&RUN, because a lot of times that's where I've used it before too, but you're amplifying it so much to get it to something that's sequenceable.
Right.
Well, we run if someone's submitted an IgG control or we're running our own, it goes through the same amount of amplification as every other library on the plate, right? It's not like we're amplifying it to the point where we create material. We're just, you know, subjecting it to the same exact process. And you're right. I mean, it's often nothing there, and so you're kind of measuring like noise in the sequencing data.
Yeah, we manage that by pooling. I mean, it goes through the same amount of PCR. It's usually the lowest yield sample, but then we just adjust our pooling, and it doesn't always get as many reads as everybody else, but yeah, it's the best we can do.
Yeah, they often wind up as like a sample that you have to add the entire amount of library to hit the, you know, amount of nanomolars that you're trying to get into that pool, and even then you won't recover very many reads. And I usually see that as like a positive result, right? Is, you know, if everything else has 11 million reads and your IgG control has 9,000 or something like that, then that just tells you you have a very low background.
But I guess there's the lack of ability to control for what's just not sequenceable in the genome if you don't have an input.
Can you repeat the question?
So my thought of an input is there's also regions of the genome that just don't sequence, so you don't know the difference between something that's not sequenceable and a peak that's lost kind of idea too sometimes, if you're not directly comparing conditions.
It wouldn't be sequenceable in the chip or the input though, right? I mean, yeah, it's
One
tough.
would hope.
Yeah,
Totally,
yeah,
but-
it's tough.
Thank you.
Yeah, we haven't run any, like, no antibody controls for cut and run.
Great, we have about 5 more minutes of discussion time.
Maybe I can ask you, so how
Yeah.
difficult was it to adapt the ChIP-exo to high throughput format?
I will say it was done before I arrived in the lab, so I will not take credit for that. Yeah, they programmed it onto the robot before I got there. I think it depends on your platform what the adaptations are. I mean, for us, just basically adjusting recipes so that you had that dead volume in the bottom that you account for. We do have parts, I mean, it's automated, the library prep part is automated, but there are steps that are off deck that we still rely on the technicians for. And I'll say in our hands, technicians are better than robots in the data quality. It's hard to compare, but it is convenient. So there are some trade-offs there, but and there are some steps where we actually want technicians watching the robot, in case there's a bubble or something. And so, well, it's great that we can get through a lot of samples. There are also some challenges there, but yeah, I didn't implement that. But with our change in sequencing in-house, actually, that coming off deck from the robot to the next steps has become easier. That I've helped to do. Do you do automation? I know you said you had 96-well format.
Adapting. We do everything. Actually, we have adapted all our assays like CHIP, CUT&RUN, and everything in high throughput format, but we are still doing manually. We don't have a liquid handler yet.
It's actually, our technicians actually really like doing it in 96-well plate, just multi-channel pipetting, better than they like operating the robot. It's fast.
Yeah, I applied for S10 grant last year, so yeah.
I think with the cut and run, the robot is really critical for processing because it's all on magnetic beads, right? So the washes on the beads can all happen at once. There's no temporal delay as you wash from, and there's lots of washes that happen, right? So as you're moving across a plate, there could be temporal, a gradient across a plate. So I think, being able to have the robot just to take care of some of those steps is totally critical to doing it in a high throughput format.
Great. Any last topics you wanted to touch on? We have a couple minutes. Well, I want to thank you guys for coming. I'm so glad this worked out. It's really been a lot of fun. Thank you to the audience for participating. We're happy to talk afterward. This has also gotten the gears turning, so I think if anybody's interested in the idea of epigenomics research group or other things, let's chat. Let's get the ball rolling. Great. All right, thank you so much.
Thank you,
Thank you, guys.